afternoon, you all come back here, okay? Because according to our experience in the past. And also, after all the talks, we have a wonderful welcome back TGIF hosted by, again, MSAC, okay? What else? Oh, please mute your cell phone, okay? Don't forget that. So this year, we have Professor Brett Rosenheim kick off the series, and his talk is about DOC in the Gulf of Mexico. It doesn't work. That's why it took me a while to. Plug it. Yeah. Page up, down.
Oh, this controls the slides too?
Yeah.
Okay, good.
Forward.
So you don't have to use both of them?
Right.
Great. Thank you. All right. Okay. Well, thanks everybody for coming this morning, and thanks especially to Tim Conway, who switched his slots with me at the last minute yesterday, because I have to go to Tampa for lunch. Now, for those students who've had me before, you've had me in both chemical oceanography and geological oceanography, but the talk that I'm going to give today, I think will hopefully serve as a tryout so that I can also teach in biological and physical oceanography. We'll see if they like the questions that I've come up with by the end. Maybe they'll give me a shot. But that's where we're going to go with this. But I'm going to talk about some research that I've been doing over the past since about 2014 in the Gulf of Mexico, and it's related to the oil spill funding. We led 3 cruises to the Gulf of Mexico, and did a lot of different work, but what I want to focus on today is dissolved organic carbon and the water column. All right. So, oops, is it back? There we go. I put my name on the first slide. You know, if you don't like what I'm saying, that's my fault, but the people who really helped make this, anything that you liked in this work are listed here. A lot of these people were on our final cruise in 2014, and the ones in bold have been involved with me in writing a paper that contains the results that I'll be talking about today. But a lot more people were on our earlier cruises too, and a lot of hard work went into this, and they've helped make this a good project. So first, I just wanted to start with a quick review, especially for newer students who haven't been through this. What is DOC and why is it important? It's probably the most important part of that question, right? So DOC, we're talking about a water column property, carbon in the water column, and if we want to look at what this is, we start off with our Niskin bottle and take our water sample. And there's all different forms of carbon in this water sample, and the first thing that sort of the biggest part of this carbon is the dissolved inorganic carbon. And to get rid of this, you simply add acid and you emit CO2, because all of that's in basically one chemical species, and you can get rid of it by changing the pH in the sample. After that, you still have carbon in the water, and it's of the organic variety, and at this point we would filter the water. You don't necessarily do things in this order. Usually you take one sample for DIC and one for the other, but the water that you pass through the filter, whatever stays on the filter is our particulate organic carbon, and whatever passes through the filter is our dissolved organic carbon. So this is a bit of an operational definition, because different people, different techniques use different filter sizes, and that means you're going to get slightly different differentiation between the POC and the DOC, right? Let's see. The other thing to point out is that this is a very big reservoir of carbon in the DIC. The DOC is not as big, so one might ask for that second part of the question, why is it important? Well, it's still a very big part of our carbon cycle, right? Carbon in the atmosphere is important, and it's about the same size as our reservoir of carbon, of dissolved organic carbon in the oceans. Furthermore, dissolved organic carbon shows an interesting trend here. Now, these are isotopic values. This is a radioisotope carbon-14, and it's related to age, although it's not plotted as years here, it's plotted in depletion. But basically you have younger values on this side, older values on this side. This is the normal trend for DIC. It's to get older as we go into deeper water. And of course, when we get into the deep ocean, things kind of calm down and straighten out, and everything's sort of the same age, okay? And I kind of put age in quotes there because it's not directly related to just the age of the water, but we can also use this as a tracer if we know some independent ages. Now, DOC, on the other hand, has the same form, but it's all older than DIC, usually in any one part of the ocean. And this has been known for a few decades now, but it's sort of a conundrum. And the question is, what makes it old, and what exactly is it? So this age is about 6,000 years old, at about -500 here. And that's on the magnitude of sort of the residence time of, or the overturning time of the oceans, okay? So if you take this sort of curve here, this is a nice depiction of this curve, and we kind of break the dissolved organic carbon into different pools here, and there's a labile pool, which is sort of biologically reactive. It's cycling in and out of organisms pretty rapidly, and this is on the younger side of things. Then you get into semi-labile and refractory. And the refractory DOC is kind of has always been the big mystery. What is it? Where did it come from? And is it even at steady state? Is it building up? Is it going away? Sorry about that. You notice there was no chemical information when we break that down into labile, semi-labile, and refractory. And that's because there's such a broad myriad of chemical forms in the DOC. It's very difficult to separate these out. And I'm an isotope person, and isotopic approaches have been very important in recognizing some of the DOC signals. In fact, a recent paper that I read had this quote about how it's basically impossible to tell all of the different biochemical and chemical forms in the DOC, but we're still trying, and that's always struck me as odd. It's kind of a really hard nut to crack, and that's why we've been able to figure a lot out with isotopes, but there's also sort of a low ceiling with isotope approach, because if you do want to know what exactly the DOC is or where it's coming from, you really have to know something about the chemistry of it, but it's very diverse. And this is a cartoon of the same thing. We have, instead of breaking it into refractory, labile, and semi-labile, we can also look at low molecular weight and high molecular weight, hydrophobic and hydrophilic. So these are sort of classes of chemistry, but we're not getting down to compound levels, and we can't relate these chemicals to processes very easily. This is from a paper by Prosper Zig et al. That just came out in GRL. And it just shows, when you see something like that, it's complexity, right? There's a lot of different arrows and a lot of different potential pools. But one of the most disputable things has been terrigenous input of DOC being one of the sources of old DOC and accumulating in the ocean. Of course, you have other sources, hydrothermal vents. Those of you who've taken geologic oceanography know about serpentinization, formation of organic molecules, even abiotically. And then you have things like methane seeps, right? So it's a really interesting system here. And what I want to focus on today are radiocarbon and C13 profiles from ocean DOC that we've taken in the Gulf of Mexico. And you can see this is setting up, it was not related to the Gulf of Mexico, but you have methane seeps, you have rivers, right, like the Mississippi River, and you have complexity. And at the time we started making these measurements, there were only a handful of ocean profiles of DOC C14 in the world's oceans. And we now have the densest set of profiles in terms of space and some of the most dynamic that I'm going to get into as we go along. But I want to make one detour to kind of give this more of a scientific point. I'm going to ask a question that kind of drove all of our research that was related to our oil spill work, and that is, in what archives are signals of the Deepwater Horizon oil spill recorded? It was an extreme event in our environmental capacity. Is it a geologic event? What archives are going to store information about this event? Okay. And of course, a lot of our work revolved around sediments. Okay. So obviously sediments serve as an archive. They always have. And one of the questions is how long will any signal from the Deepwater Horizon persist? So just to kind of give a background on the isotopic work we did, we took a lot of sediment cores during our cruises as well as water column samples. We compiled baseline data. These are stable isotope data from sedimentary organic material that were published before the Gulf oil spill. And you can see that oil, these triangles or seeps, they have a much different color or isotopic value than typical organic matter. And that gives you a little bit of leverage, right, to see if oil has been incorporated into the sediment. Radiocarbon gives you even more because it's not even shown on the scale, but oil would be about -1,000. Unfortunately, there's just not a lot of baseline data to set up sort of an isoscape to try to discern the amount of oil that has been incorporated into the sediment. So we started off using stable isotopes, but knew that there was a lot of power in the radiocarbon approach. And then we started working with Jeff Chanton, who works within the C-Image group, and David Hollander. And he came up with the nice idea that since sedimentation rates are so high in the Gulf of Mexico, the radiocarbon values from the organic material in the sediment would more or less be a flat line once you get below the signature of oil, okay, a vertical line. And that's because the sedimentation rate here, this might only be about 20 years of sediment depth. And in 20 years, the half-life of radiocarbon is on the magnitude of 5,000 to 6,000 years old. So there's not going to be a big change in radiocarbon values over that 20-year time span. But when you get to the tops of these cores in the Gulf of Mexico, things go towards older, right? And they shouldn't, because sediment on top should be younger, right? So there's a reversal, and it's far more extreme than the sort of constant values that we saw below that effect. So we're able to say that these values have been affected by oil, and these values were our baseline values since before the oil spill. Okay? And furthermore, when you go to a seep site, you see far more negative or older values there because you have a lot more incorporation of oil. So this type of profile is what we're looking at for oil contamination. And these were just, we're trying to avoid the seep sites because we want to find out how much of this oil was deposited in the sediment. And we were able to come up with a nice isoscape using C14. Even though in the previous slide I showed there wasn't a lot of baseline data, Jeff had the really nice thought of using down core data as baseline. And we came up with this sort of heat map. The hotter colors are more negative or older values or more petrocarbon. So anytime we get some of the oil or methane that somehow gets incorporated in the sediment, we're calling that petrocarbon. And from this sort of heat map, we can come up with a budget using mixing models, okay? And this is, sort of a nice view of a mixing model. This is how I think of them, but, sort of like a balance or a seesaw. And if we want to keep this horizontal, but we have different weights, so to speak, these are the isotopic values, that's oil, and this is the -200, which is where we have that profile straightening out in the sediments. If we measure a value that's not either of these end members, we get something in the middle, then we can simply use a two end member mixing model to calculate what percentage of it is oil, what percentage of it was our background sedimentary organic matter. And in this case, with these numbers, this would turn out to be about 37% oil. So we do that in all those different points on our heat map, and we come up with a budget. And we found out that somewhere between 3 and just about 5% of the oil released during the Deepwater Horizon spill was residing in sediments. And what's nice about this isotopic approach is that it's irrespective of chemical changes, right? We didn't focus on the chemistry or one chemical compound within the oil. Oil is a complex mixture of compounds. So, and this is a nice, a tractable number that we can add to the budget. So I want to get back to this question, though. In what archives do we see a signal of the oil spill? And this is where we get back to DOC, because I also thought that since this is an event, this is an environmental event, it's happening more or less right now in a few years, we might be able to see something in the water column. So we've seen something in sediment, we've been able to use that. What about the water column? And this is kind of a crazy idea, because you don't really think of the water column as an archive, right? Geologists don't, at least. So. And here's the premise of this, is that we have a some sort of a spill event at depth. We're releasing oil bubbles that are hot but compressible. We're releasing methane bubbles. There is water movement at depth, but it's probably much less than wind-driven movement at the surface. We do get fallout plume and we get oil back into the sediments, but we have methane that dissolves into the water column, and we have oil that resides in the water column, okay? And here are our pools of carbon in the water column that I brought up earlier. We have DIC, so if we remineralize any of this oil or methane, that means we would use oxygen and we would oxidize that, and we would end up with basically CO2 dissolved in water, or DIC, okay? So I wanted to check that. There was evidence right after the spill of POC, so that's falling through the water column. Even though we get that in our water sample, it's not really a water property. POC is being generated in the surface ocean, falling through the water column. But right after the spill, it was noted that there was a petrocarbon signal in the POC. That would be short-lived. But then the other one that nobody had really checked on, DIC and DOC, both POC and DOC, you have biosynthesis that could potentially incorporate a POC, or a petrocarbon signature into these two pools. Okay. So I want to go back to the mass balance, right? And the problem is DIC is such a big reservoir that the little piddly oil spill doesn't produce, even if you remineralize it all, it doesn't produce enough carbon to flatten out that mixing model, right? So that's the elephant in the room is when we're measuring DIC, it's just too big of a reservoir to be affected by the oil spill, okay? And here's our results. We measured anyway. We're stubborn, but but you can kind of see that when you get down to the plume depth of about 1,000 to 1,200 meters, everything kind of lands on top of each other. And this is pretty typical of the Caribbean too. Interestingly, the most variability you get is in the surface ocean. This is probably loading due to the river, and that's a whole other story. Just the typical dynamics of the Gulf of Mexico, right? You don't normally see that much variability within a short spatial scale as in the Gulf of Mexico. So in our final cruise, this is the map of our cruise, pretty small area. We decided we were going to follow the plume out of the Mississippi River. We took out of Terrebonne Parish here at the Lumcon station. We went up into the river in Southwest Pass. That's the major outflow of the river. We had a site in the river. We came back out, and with the help of Frank's group, Frank Mueller-Carger's group, and namely Daniel Otis, we were able to get KMZ files beamed out to us from salinity model outputs and satellite imagery to figure out where the plume is. Normally the plume comes out of Southwest Pass and goes this way. This is salinity plotted here, and you can see that during our cruise it was sort of an oddball time, and we didn't see much plume coming out that way. In fact, the plume was going to the northeast, which is strange. It's a very small minority of the time that happens. So we followed the plume out. So we followed this water mass as it's coming out of the river and mixing with the ocean. We wanted to see the dynamics of that. That's sort of one of our end members. And then we proceeded to go down past the well site marked by this star into the loop current and back around to older plume water that was residing on the shelf here. So that was our cruise path. And we had primary sites, these P sites, and we had secondary stations where we didn't do full depth. So we want to measure the carbon-14 ratio of DOC. Each measurement takes 11 hours. It's a lot of work. So we couldn't take all of these profiles, and that's why there's so few of them that were in the ocean at any one time. But this is pretty, to have this many in a small area was really neat. Well, we hope so at least. And this is what we saw. So I want to focus your eyes over here. Oh, by the way, A20 was the closest place where we had a DOC C14 trend, and that's shown by these blue lines here with smaller dots. And what we have is we have the concentration of DOC, we have the C14 of the DOC, and we have the C13. And some of you who've already taken chemical oceanography probably recognize these plots, right? Yeah, I'm seeing some nodding and some of this too. Anyway, we see at depth an increase in DOC, okay? That's not in our loop current site at P4, and it's not in the Caribbean in A20, and it's not in our secondary site at S4, okay? That same increase in DOC is a very large negative excursion in the C14, okay? Now remember, the C14 of oil or methane from the spill would be negative 1,000, okay? And we can start to think about this as a mixing model. We have an increased concentration, we have a decreased signature. We can basically calculate what was added, what was its isotopic signature of what was added, all right? And at the time when we measured these were the most negative DOC C14 measurements made in the world's oceans at that time. So these are pretty interesting measurements to stumble upon. And then you can see that C13 gets very negative as well, but not in the same fashion as C14, and we'll get back to that in a second. So, so what's happening here? Now this is where I'm trying out for biological oceanography, and you guys can let me know if I'm doing a good job. So we have droplets of oil and CH4, right? And we have stuff that comes in and eats it, and then it leaves. Okay, good. Whew. All right, I passed. It does do some remineralization, right? Because right after the spill, there was a drawdown of oxygen in some measurements that, is probably formation of CO2. But again, it's just, we can't get to that mass balance because DIC is such a big pool. But we do leave behind some biosynthesized molecules in the water column. So in the paper that we just put out last week, microbial transformation of mesopelagic deepwater horizon oil and methane into a natural population of recalcitrant DOC molecules is what we're interpreting this signature to be, all right? And we can get back to our mass balance. Now we have about negative 200 is the DOC from the other sites that didn't show this signature. We have the oil here, and we calculated if the plume, if we use the volume of the plume or the area of the plume right after the oil spill and assume that this was happening all throughout that plume, we only have one profile where it's happening, so this is a big extrapolation. We find that we could get conservatively, and this is actually pretty conservative based on the concentrations we use and stuff like that, 10 to 16% of the released hydrocarbon transformed to DOC at these two depths, 1,000 meters and 1,250. Okay, now think about that compared to the sediments. Sediment archive was kind of a given. You know that there's going to be oil in, and the question is how much. With the water, this was kind of a Hail Mary. The water column you didn't think of as an archive, but here we're seeing, potentially a lot of petrocarbon, a lot of the spill residing as this population of DOC molecules, okay? C13. So now we have an end member here with methane of negative 60. We have the oil was measured about negative 27. There's less dynamic range in this. That's why stable isotopes, radiocarbon is more, is preferable to stable isotopes. But with methane stretching things, it gives us a nice scope to work with. So at each plume, at each depth, at 1,000 meters, we found that the petrocarbon signal was about 40% methane and 60% oil at 1,000, and only about 10% methane and 90% oil at 1,250 using these end members, because we have that bizarre sort of decoupling between C13 and C14. That's what we're interpreting it as. Now, we also have these sites, which I didn't talk about, and these are our P1 river site, and this is following the plume in P2, and then P5 is the older plume water. Okay, and there's a lot going on here. These are big numbers. You can see, well, let me just show you our sort of cartoon that we've drawn. I called this talk Deciphering DOC in the Gulf of Mexico. I'm just going to mention this, but it's really interesting. In the Mississippi River, we see an output of young DOC. And the thought is that's one of our sources of old refractory DOC in the ocean. And we actually see that this color change is to represent what we talk about in the paper. And that's a gradual transformation. Actually, it's not so gradual, right? It's over the course of days, a transformation of that DOC into a different marine primary produced pool. So it's kind of pulling the terrestrial DOC out and changing its isotopic signature, during the days that we were on this cruise. And that's pretty, compared to open ocean work that's been done so far, this is a pretty interesting finding. We have, of course, the plume water, and then we have the water from the Gulf of Mexico at depth is actually a little bit radiocarbon younger than the water from the Caribbean, so there must be actually a source of young water. One of the early hypotheses we had when we first got our negative DOC signal in C14 was that maybe the whole deep Gulf, since it's a sealed basin, is just full of DOC from natural seeps and everything. And of course, the next data set that we got from further south in the loop current didn't show the same signal. So there's actually another young source, too, that we haven't identified. So, of course, I'm going to shoot myself in the foot for the physical oceanographers. They're probably not going to want me teaching that class because, this is the big question: is how we made these measurements in 2014. The spill was in 2010. How do you still have a signal at this depth in this part of the Gulf of Mexico 4 years after the spill? And is it still there today? That's the question. And I think that this question is fair game because we don't know a whole lot about currents once we get off the surface or away from the bottom of the ocean. And this is sort of right in the middle of the water column where we're seeing this signature. So at that, I'd like to open up for any other questions, or if anybody has a shot at this question, feel free. And thank the people who funded this. Through GOMRI, I was a member of the Carth Consortium, and Brian Roberts, one of the authors, was in the Coastal Waters Consortium.